Welcome to the Deep Dive, your express lane to mastering complex research. Today, we are really getting into the weeds on muscle, specifically the giant protein titin.
It's a fascinating one. Titin is, well, it's the biggest protein we know of. It acts like a molecular spring inside your muscle fibers.
Right, the thing that gives muscle its elasticity.
Exactly. But when it goes wrong, due to mutations, you get these really severe muscle diseases. They're called titinopathies, muscle wasting, weakness.
And we've known for a while that titin is sort of, well, dual purpose, haven't we?
Yeah, absolutely. On one side, it's this massive structural element. Think of it like a suspension cable holding the muscle's contractile unit, the sarcomere, together, connects the Z line to the M line.
The anchors, basically.
Pretty much. But then it's also a signaling hub. It binds to loads of other proteins that, regulate whether the muscle grows or shrinks, hypertrophy or atrophy.
Okay, but here's the rub, Ray. The big question researchers have wrestled with. How do you actually separate those two jobs, the mechanical part and the signaling part?
That's been the core problem. The hypothesis was always that the mechanical tension itself, the actual physical force titin handles, might be the critical factor in disease. Losing that tension could be the trigger.
But you couldn't test it directly.
Not easily, because if you just use the old school gene knockout approach, delete the titin gene entirely, you lose everything. The structure, the signaling, all of it goes at once.
Makes the results really hard to interpret. You don't know what caused what.
Exactly. It's a classic issue in biology, that chicken and egg problem. Lots of proteins have this dual role, sensing force and signaling. Things like talin, lamin, dystrophin.
So the sources we're digging into today describe, well, a really clever way around this. They set out to test that specific hypothesis. What if you only mess with the mechanical function? Leave the protein there, but just cut the cable.
And the answer they got is, frankly, quite profound. They developed this method they call mechanical knockout, or MKO.
MKO.
And using it, they showed that simply losing titin's ability to bear force, just stopping that mechanical tension, is enough. All by itself. It triggers rapid, severe muscle breakdown, mimicking the human diseases really closely.
Wow. Okay, so that loss of tension is the trigger.
It certainly looks like a primary trigger, yes.
We absolutely need to unpack this MKO method then, because it sounds like it could be useful way beyond just titin research.
Oh, definitely. It's potentially a game changer for studying any mechanosensitive protein. So first, let's just appreciate titin's scale again. It's huge, up to 3.8 megadaltons.
Which is just enormous compared to an average protein.
Ridiculously big. And it spans half the sarcomere, remember? It's the main reason your muscles have that passive stiffness, that resistance when you stretch them.
Okay, so if you want to cut the spring without removing the whole thing, you need something precise, like molecular surgery.
That's a good way to put it. What the researchers did was engineer mice called TEVs, TTN mice. These mice have a tiny specific modification in their titin gene.
What kind of modification?
They inserted a recognition site, like a little landing pad, right into the I-band region of titin. That's the elastic part, the bit that stretches.
A landing pad for what?
For a very specific molecular scissor. It's an enzyme called tobacco etch virus protease, or TEVP. It's a tool biologists use a lot because it's highly specific.
Right.
It only cuts at its designated recognition site.
Okay, so you've got the target site in the mouse titin, and you've got the specific scissor. How do you get the scissor into the muscle?
They used a common delivery system. A virus, specifically an adeno-associated virus vector, AAV6. Think of it like a harmless delivery van. They load the instructions for making the TEVP scissor into the AAV6.
Like inject it into the muscle.
And the muscle cells take up the AAV6, read the instructions, and start producing the TEVP scissor inside the cell.
And that scissor finds the landing pad on titin SNP.
Exactly. It cuts the titin protein right at that engineered spot in the I-band. And here's the crucial part. This achieves the mechanical knockout.
Because the protein chain is broken.
Right. The polypeptide backbone is severed, titin goes slack, it can no longer transmit force along its length. But, and this is key, the total amount of titin protein in the cell doesn't really change.
So the protein's still there, it's just been neutralized mechanically.
Precisely. It's present but functionless in terms of tension. A really elegant piece of engineering.
And they showed it worked.
Oh, yeah. Within just 7 days of delivering the AAV6 vector, they measured the muscle fibers. In the homozygous MKO mice, that means mice where both copies of the titin gene were targeted, the passive stiffness plummeted.
How much?
A drop of 35% to 65%, a huge loss. It was clear proof the spring had been cut.
Okay, so the mechanical integrity is gone.
Mm.
What happens to the muscle tissue itself? You said it was rapid and dramatic.
It really was. This wasn't some slow, gradual decline. They saw significant muscle atrophy starting very quickly.
How quickly and how bad did it get?
At the whole muscle level, by just 21 days after injecting the virus, the homozygous MKO muscles, the ones with the fully cut titin, had lost a massive 38% of their mass.
38% in 3 weeks? That's huge.
It's incredibly severe atrophy. And functionally, those muscles were much weaker too. They measured the maximum force the muscles could generate, and it was significantly reduced compared to the control muscles. So structural loss and functional decline went hand in hand.
And where does the breakdown start?
Yeah.
Right at the sarcomere, where titin lives.
That's what they saw. Very early on, just 3 days after the key EV started cutting, they could see the severed I-band part of titin starting to, well, recoil.
Recoil.
Yeah, like snapping back towards the Z disc, the anchor point. Imagine cutting that tensioned cable, the cut ends would immediately pull back. That's what they observed happening with titin.
And this was happening before the whole sarcomere fell apart.
That's the really interesting part, yes. The sources emphasize this recoiling, this clear sign of mechanical unloading, was visible while the overall sarcomere structure, like the Z discs themselves, still looked relatively normal, still intact.
So the loss of tension comes first. That's the initiating event.
It strongly suggests that, yes, the mechanical failure precedes the larger structural collapse. Because by day 7, just 4 days later, things looked very different. The organized, striped pattern of the sarcomeres was basically gone in most of those affected muscle fibers. Total disassembly.
Wow. But then there was something unexpected about the cells themselves, right? They didn't all just die off.
This was a really compelling finding. You'd think with such catastrophic internal damage, the muscle fibers would just degenerate completely. But they found a surprising number of these titin MKO fibers, the ones without organized sarcomeres, actually survived.
Survived. How?
Well, they shrunk dramatically. They became these sort of disorganized sarcomere-free husks. But they remain viable, sticking around for months. They track them up to 92 days post-infection.
That kind of challenges the idea that the sarcomere is absolutely essential for immediate cell survival, doesn't it?
It does raise questions. These fibers are clearly dysfunctional, shrunken, but not immediately eliminated. The study suggests that eventually, over time, these damaged fibers are slowly cleared out and replaced by new, healthy fibers through the muscle's natural regeneration process. But it's not an instant cleanup.
Okay, let's zoom out a bit to the tissue level again. You mentioned the MKO muscles looked like human muscle disease. What specific features did they see?
They saw pretty much the classic hallmarks of severe myopathies: widespread muscle fiber atrophy, the shrinking we just talked about. They saw increased fibrosis, which is scarring, basically collagen building up between the fibers.
Making the muscle stiff and less functional.
Exactly. And they saw major problems with mitochondria, the powerhouses of the cell. They were mislocalized, clumped together abnormally.
And what about the myonuclei? You mentioned those earlier. They're usually right at the edge of the fiber.
Right. Normally they are anchored peripherally, but in many muscle diseases and under stress, they move towards the center of the fiber. This study showed something quite profound. Titin's mechanical integrity seems to be essential for keeping those nuclei pinned to the edge.
How so?
Because they saw myonuclei starting to move inwards, becoming internalized as early as day 3 after inducing the titin cleavage.
Day 3. That's even before the sarcomeres fully disintegrated in many fibers.
Precisely. It suggests the nucleus itself, or the machinery that positions it, is somehow sensing that loss of titin tension very, very quickly. It's not just a consequence of the whole fiber falling apart later. It seems to be a more direct response to the mechanical change.
A direct link from the spring to the cell's command center. That's fascinating.
It really is. And other signs of distress appeared just as fast. Desmin, which forms an internal scaffolding network in the cell.
Sort of holds everything in place.
Yeah. That network started to mislocalize very early, too, again around day 3, even preceding the nucleus moving. And the mitochondria started forming aggregates almost immediately, leading to this characteristic halo ring pattern by 21 days. It's a rapid dominant effect once tension is lost.
Okay, that's the severe picture in the homozygous mice, where most of the titin was cut. What about the heterozygous ones, where the cleavage was only partial?
That's a crucial part of the study because it provides a bridge to perhaps less severe human titinopathies.
Oh.
In the heterozygous mice, they achieved about 24% titin cleavage, much less than the homozygous.
And the resulting disease was milder.
Milder, yes, but still significant and very revealing. They didn't see that catastrophic mass loss, but they did observe important pathological features. For instance, a reduction in the number of large, healthy muscle fibers.
Okay.
They also saw some centralized nuclei and Desmond mislocalization, though less widespread than in the severe model. And importantly, when they stained the muscle tissue histologically, they found structures called cores. Yeah, these are specific areas within the muscle fiber that lack oxidative enzyme activity, visible with stains like SDH. And these core-like structures are a well-known feature in certain human congenital myopathies, including some linked to Titin.
So the partial mechanical knockout directly recreated a specific feature seen in human patients.
It strongly suggests that level of Titin dysfunction, that partial loss of tension, is enough to cause these specific pathological changes.
And did they look at the gene activity? Did the molecular signals match up too?
They did. They analyzed the gene expression patterns in the MKO muscles and found a really striking overlap with a classic mouse model of muscular dystrophy called the MDM mouse.
How much overlap?
Depending on the comparison, between 47% and 68% of the genes that changed expression in the MKO model also changed in the MDM model. Things like increased fibrosis pathways, decreased mitochondrial activity pathways.
So triggering disease purely through mechanical failure activates a very similar molecular distress program as a genetically inherited muscular dystrophy.
That's what the data strongly indicates, yes. It reinforces the idea that this mechanical pathway is central to the disease process. Which brings us nicely to those molecular signals themselves. If Titin acts as a signaling hub, what happens when it suddenly goes slack? What alarms does it set off?
The study pinpointed one very early responder, didn't it?
Yes, a protein called MERF1. That stands for muscle-specific RAN finger protein 1.
MERF1. I know that one. It's a key player in muscle atrophy, right? It tags proteins for destruction.
Exactly. It's a ubiquitin ligase, part of the cell's protein disposal system. When muscles are wasting, MERF1 levels usually go up, and in the MKO muscles, its expression absolutely skyrocketed, starting as early as day 3.
Day 3 again. So before the muscle mass loss really kicked in, before the sarcomeres were totally gone.
That's the critical insight here. The cell seems to initiate the demolition process, ramping up MERF1 before the major structural damage is even complete. It's like MERF1 is responding directly and immediately to that loss of Titin tension.
So it's not just cleaning up the mess afterwards, it might be actively driving the breakdown in response to the signal.
That's certainly what it looks like. It suggests MERF1 is a key, very early mediator in the pathway linking mechanical unloading of Titin to the subsequent atrophy.
Were other stress signals activated later on?
Yes. From about day 7 onwards, they saw other changes kicking in. Increased activity of an enzyme called calpain-3, that's a protease, another protein cutter that's known to interact with Titin.
Okay.
And they saw increased levels of several chaperone proteins like ANKRD1, HSP27, and alpha-beta crystallin.
Dish chaperone, those are involved in managing stress and trying to keep proteins folded correctly, right?
Exactly. It looks like the cell is trying, desperately perhaps, to manage the stress and maintain the quality of the proteins that are still functional, even as things are falling apart. It's a full-blown stress response.
Given how central MERF1 seemed to be, did they try blocking it?
They did. They used a known MERF1 inhibitor, a compound called Biomed 205. The hope was if you block that early demolition signal, maybe you can prevent or slow down the muscle wasting.
And did it work?
Unfortunately, no, at least not in the severe homozygous MKO model. The inhibitor wasn't able to bring MERF1 levels back down to normal, nor could it prevent the downstream protein ubiquitination or stop the muscle fibers from shrinking.
So just blocking that one pathway wasn't enough when the core mechanical problem was so severe.
It suggests that the consequences of completely severing Titin's force transmission are just too profound, too aggressive. Once that central cable snaps entirely, simply telling the demolition crew to pause isn't sufficient to stop the building from collapsing. You likely need more comprehensive interventions. So let's try and synthesize this. What this research really achieved, quite brilliantly, was isolating Titin's mechanical role.
Separating it from its other signaling functions.
Exactly, and by doing that, they demonstrated pretty definitively that just stopping the force transmission across Titin, just cutting that spring, is enough. It's a direct and incredibly rapid trigger for severe muscle disease.
And the damage isn't just contained within the sarcomere, it spreads out fast.
Immediately.
Right.
We saw effects propagating to the myonuclei, to the mitochondria, activating widespread stress responses almost instantly. It really underscores how fundamental that mechanical integrity is.
It solidifies this idea that Titan's ability to bear tension is, well, non-negotiable for muscle health, like an Achilles heel for the entire tissue.
That's a very apt analogy. Whether it's engineered, like in the study, or happens because of gene mutations that truncate the protein in human titinopathies, losing that tension seems to be a primary driver of the pathology.
Which brings us back to that difference between the severe homozygous mice and the milder heterozygous ones.
Right. The heterozygous mice had about 24% of their titin cut, leading to a milder myopathy-like state. The homozygous mice had closer to 68% cut, leading to catastrophic atrophy.
That difference, that dose-response effect, raises a really interesting final thought for you, our listeners, to mull over.
If the severity of the disease seems linked to the percentage of titin that's mechanically compromised,
Is there potentially a critical threshold? Could there be a specific point, say, somewhere between that 24% and 68% cleavage level, where the muscle tips over from managing a mild dysfunction into a state of rapid, irreversible decline?
and thinking therapeutically, if such a threshold exists, could future strategies aim not necessarily for a perfect fix, but perhaps just to keep the level of functional tension-bearing titin above that critical line? Could preserving, say, 50% or 60% of titin's mechanical integrity be enough to prevent the most severe outcomes?
A fascinating question for future research and potential therapeutic design, focusing on maintaining sufficient tension rather than just presence.
Indeed, lots to think about.
Absolutely. Well, thank you for joining us for this deep dive into titin, tension, and why pulling its weight is so critical for muscle. We'll catch you on the next one.